Well, good morning again. It's a pleasure to have the opportunity to moderate this session on autoimmune diseases and applications of cell and gene therapy to this particular area of biology and medicine. My name is Farzan Farzaneh. I'm the CEO of Viracell Biologics. And Viracell is a company focused in precisely the sort of areas that we will be addressing today: development of new technologies and their application to the rapid transition into the CDMO space, through the CDMO into the clinical application space. And as gene therapy expands and becomes applicable not only to the malignantly transformed cells and oncology space, but into much more prevalent disorders such as autoimmune disease, issues relating specifically to technology in the production of the vector, in the safety of the vector, and as Sumanta and I were speaking a little earlier, all manner of auto and inducible regulations for better control and for better safety. So without further ado, I would like to introduce Ben Duys, who is the VP of Regulatory Affairs and Quality at Kyverna Therapeutics. Thank you, Ben.
Thank you very much. Really excited to be here and really start to explain about Kyverna and what we've been working on. Kyverna's been in business for about 5 years, and we've been focused on autoimmune diseases the entire time. So there's been a lot of great press recently about us. We just had a great IPO. We've been sharing some clinical data that you'll be able to see later today. But it looked like an overnight success. It's taken us 5 years to have that overnight success. So really excited to share why autoimmune diseases are so important to be focused on. About 8% of the US population has an autoimmune disease: rheumatoid arthritis, multiple sclerosis, Sjogren's disease, lupus. There are a lot of very small niche indications as well that we're targeting, and Kyverna's products could be effective in 40 to 80 of them with just a few products. So the background data that really got us excited about getting into a CD19 CAR T therapy was looking at the B cell depletion in patients with a pretty rapid CAR T expansion and then a quick drop off. And if you look in the upper right graph, you can see that there's a real strong activated memory B cell depletion, and then a reconstitution with memory B cells that are not triggering the autoimmune indication again. So our construct is fully humanized. We selected it from, we licensed it from the NIH. It demonstrated a much safer safety profile in oncology patients, and so we wanted to move forward with the safest construct we could find available. And we chose CD19 because it really does cover the whole B cell gamut that we wanted to be really wiping out as quickly and efficiently as possible. We didn't want to leave anything lingering that would give problems, but still sparing the long-lived plasma cells, so that we're still maintaining people's vaccines. They're not being completely wiped out of their historic immunity. Concerns around safety for a non-terminal oncology patient is something that comes up quite frequently when we're talking about autoimmune diseases. What about the safety? We really think that the safety profiles theoretically should be lower when we're treating autoimmune patients because they don't have the same tumor burden, and they don't, and we're able to use a newer, safer construct than some of the marketed products. So we're just not expecting to see that same safety profile, and we'll see data on that. So we've been very active and very enthusiastic about and getting into the clinic. We have 2 studies in lupus nephritis, one in Germany, one in the US. We've also initiated, we've cleared INDs for systemic sclerosis, myasthenia gravis, multiple sclerosis, and that's going to be a pretty robust study. We're going straight to phase 2 there. So we've been collaborating with the FDA, and these are just the first 4 indications. We can just keep going down the list of autoimmune diseases that should theoretically benefit. And that's why we're so excited, and we're really digging in and getting as active as possible. We've done a lot in the United States, and we're pivoting and doing more work in Europe this year, and excited about making that move. We're not just relying on that one product. We have an allogeneic version that we've been partnering with Intellia with that is moving into the clinic soon, and other treatment modalities and second-generation products as well. So let's dig in. We'll skip over this for time, but let's dig in and really look at what the patients are doing. As of the end of last year, which is what the lawyers say that I can talk about, we had apheresed 22 patients, dosed 14, and had more than a month's follow-up in 13 of those, and both in clinical trials and as well as initiated investigator-initiated trials and compassionate use. So what does our data look like? We were getting rapid expansion, just as you would expect. So the bold lines are our patients that we've been treating. The faded outlines are the oncology patients that were treated with the same construct with the NIH. So you can see very consistent cell expansion and decline in the way that we're really wanting to see. We're seeing B cell counts that are dropping quite rapidly, and then coming back. And most importantly, is for our lupus patients, is we're seeing the urine protein, the creatinine ratios dropping quite rapidly and staying down, even if we do see some B cell replenishment by the bone marrow. What does it look like in a myasthenia gravis patient? I'll focus on the right side here. What we're seeing is one of our patients being able to hold their arms up for longer. So you see some variability there, but a steady increase over the first few months. And as well, their walking mobility is flatlined at about 200 meters before they get treated, and then you see a slowly progressing improvement over time, which is really exciting. So charts on a graph look great and all that. It definitely makes the regulators happy. What really matters is what happens with the patient, and this means so much more to me than the charts and the graphs. It's important to show safety, but it's also really important to really remember why we're here is to treat these patients. And so you can see that the woman who's able to get out of a wheelchair and start walking. And that's something that we just, life-changing. Not just for her, but for her children, for her family. It makes all the difference in the world. But if you can ride your bike afterwards, go play with your kids, go to the park. And you need to remember that these people are really, they look somewhat normal and they try and live their lives, but they're missing out on so much, and their families are missing out on so much. And so 2 months later, she's able to just blend right in and take care of herself. So this is why we're here. This is what we're doing. So the product works for at least some people, maybe not everybody, but we're really optimistic about it. Importantly as well is safety. And so what's been seen in oncology patients with the CAR T therapies is some severe CRSs, grade 3s and grade 4s. What Dr. Schett's group showed and what our data has shown is no grade 3 or grade 4 CRSs or ICANS. And that may have to do not just with the safe construct that we're using, but also the lack of a tumor burden and the more robustness of the patients rather than the oncology setting. This is my favorite boring graph, and the more boring it gets, the better it gets. These are the ICANS and CRSs that we've seen in the clinic. So you can see some pretty colors on close to day 0, and then everything else is very boring, and that's exactly how I like it. So this is showing a long, durable safety profile that is completely uneventful after the first couple weeks. What else are we doing? We have a great partnership with Intellia, who's the lead gene editing company that we'd want to work with. And we're working with, we're developing that and should be filing a clinical trial application this year to be able to move into a more, well, using healthy donors is going to allow us to do a lot more, go to a lot more places. We've been talking this morning about patient access, and to be able to have a distributed product rather than relying on one patient being their own manufacturing lot, that the whole paradigm shifts when you can have a donor that's able to contribute multiple doses instead of just one. So in order to do that, we knock out, we get a TCR knockout as well as the HLA and HLA-A, as well as a pan-HLA class 2 knockout that knocks down the immunogenicity of the product, and then we're able to have the donors take care of it. We're also not sitting on our laurels with our autologous program. We have a second-generation autologous program that we're going to reduce manufacturing times. We're going to be harvesting whole blood rather than doing an apheresis process, which is quite burdensome to the patient. So we have a lot more really exciting and interesting things going on. So that's just a quick overview of what Kyverna has been working on, and I'd like to invite Savant up, please. Yeah.
Right. So we are another company in the CAR T space. Let me move on to my first slide. So we are right now the first and only CD19, CD20 directed CAR T cell therapy company for the treatment of both cancer and autoimmune diseases. I will talk very briefly about the fact that we actually, beyond that, we're also in the solid tumor space, and we'll be having a solid tumor program in early next year. So we started off as an oncology company. All our technologies come from UCLA, from the labs of Dr. Yvonne Chen and Dr. Tony Riba, so very well known in this field. And it was part of Impact Bio as of middle of 2021. I came in towards the end of 2021, helped raise the Series B, and then build a team that was beyond the research team into the clinic and the manufacturing facility that we actually run by ourselves. So we chose dual targeting as our approach because, like most therapies in both the autoimmune space and the heme malignancy space in oncology, in general, there are escape mechanisms. So you do need to have coverage and broad coverage to make sure those escape mechanisms don't arise. But we go beyond the dual targeting itself. We have an optimized in-house manufacturing process where we choose to actually select a type of cell to virally transform, and then that product is actually very differentiated compared to many of the products that are currently in the market. We are advancing an autoimmune portfolio along with our heme malignancy portfolio, which is already in the clinic enrolling patients. Our autoimmune portfolio is right now a IND cleared for lupus nephritis and non-renal SLE. And then we have plans this year in the next few months to file additional INDs and IND amendments to expand this particular portfolio. And then we are expanding a basket trial outside the US to generate data across multiple indications. So this is our general pipeline, gives you a sense of the breadth of the CD19, CD20 directed CAR T cell therapy. The first IND was cleared in January of 2023 and is currently enrolling patients in multi-center trials in aggressive B-cell lymphoma. Aggressive B-cell lymphoma, for those of you who are not aware, is DLBCL arising from transformed follicular lymphoma, PMBCL, and high-grade BCL with or without double or triple hit. That same construct and the same manufacturing process was taken into autoimmunity. Why? The study that was started with the first in the aggressive B-cell lymphoma was actually based on a UCLA study that started in the second half of 2019 and has now gone on for over 4.5 years. So we have long-term efficacy and safety data with this particular construct and manufacturing process from UCLA. In UCLA, they've had a durable complete remission rate of 73% in patients with very aggressive clinical disease in the lymphoma setting. No ICANS, despite the fact that they had very high tumor burden, and CRS was limited to grade 1 or 0. So I think there's a lot that is not well understood around why some patients get CRS and some patients don't get them, why no patients have gotten ICANS, despite the fact that in the lymphoma setting, there was significant tumor burden, and these patients did achieve CR. So we took this construct, and as soon as the paper in Nature Medicine came out in the fall, late summer, fall of 2023, we said, we have a very safe product on the oncology side. Why don't we pivot and make sure we look at what the autoimmune side could bring? So we were an oncology company, and then we pivoted about less than a year ago, into a year and a half ago into the space. In August of 2023, we got our IND cleared and have already activated sites and on the verge of enrolling patients, both in the US and outside the US, and that's what we'll focus on today. In addition to that, we do have a solid tumor program that's focused on the immunosuppressive tumor microenvironment. We are an immuno-oncology company based on cell therapy, and this looks at converting an inhibitory chemokine called TGF-beta into an activating signal for the CAR T cell, and it's coupled with a specific tumor-associated antigen. We have not disclosed this one, but we are now in IND-enabling studies with a planned study start in 2025. So let's focus on the CD19, CD20-directed CAR T-cell therapy. This is a tandem CAR. It's a single polypeptide chain, so it's quite efficient, compact, and has very good CAR expression and yield when we do viral transformation product development. It was optimized by Dr. Yvonne Chen for being CD20 binder being membrane distal, CD19 binder being membrane proximal. If you know what how these antigens are, these targeted antigens, you have to have it oriented this way. And then we use a 4-1BB co-stimulatory domain. The reason why Yvonne Chen chose 4-1BB versus another co-stim domain was because it was noted in the literature that 4-1BB generally promoted CAR T-cell persistence, as well as just a gentler rise and expansion of the CAR T-cells and lower cytokine release. So we have carried that through into our own product at Impact Bio. But equally important is the right-hand side. It's a bit of a different manufacturing process, but it's quite simple. The leukopak comes in, day one of manufacturing, we do a magnetic bead isolation for CD62 ligand. CD62 ligand is noted to be on naive T cells all the way through memory T cells. So when you isolate that as your starting material going into the manufacturing process, the product ends up quite heavily on the central memory population, with some naive T cells, virtually no effector T cells, and some effector memory T cells. So with that, you see with CD62 ligand cells in our hands and in the literature, better engraftment, improved persistence, reduced potential for exhaustion, and just generally a lower cytokine production. So here is what we have seen in our own trial in the lymphoma side that gives you a sense of the very consistent manufacturing where you have very high central memory and naive population noted on the left-hand box and whisker slide. And in the middle is the CAR expression noted. This is from patients in our current ongoing trial. So we have been very consistently being able to manufacture product for very difficult, heavily pretreated heme malignancy patients with aggressive B-cell lymphoma. On the right-hand side, you see what we're talking about when we talk about CD62 ligand cells. When we transform CD62 ligand cells versus PBMCs from these patients, we are seeing generally and very consistently lower gamma interferon production. And we see that not only in the heme malignancy side, but in each and every preclinical assay we've done with donors on the autoimmune side. And this is likely one of the reasons why we're seeing a differentiated safety profile, even with patients with high tumor burden. So what do you see in these patients? These are the first 5 patients in the heme malignancy side, and we fully expect, just like Ben presented, this will play out in the autoimmune side also. Now, you see a rapid rise in the CAR T cells, and then a slow decline so that you actually have the persistence for beyond day 28 and beyond. Because many times, the residual dysfunctional B cells are not just in the periphery. They are embedded in the tissues, in germinal centers, in protected sites like the CNS, or other protected areas in the body. So you want to get a B cell aplasia that actually gets the B cells that are hard to get. And then on the right-hand side, you actually see that result from B cell aplasia. Now, I will say, in the autoimmune side, you will have some B cells. In the heme malignancy side, these patients have gotten so many rounds of therapy, sometimes their baseline B cells are quite low. But you see this B cell aplasia, and that's what you want to see, and then the recovery will come after. So how do we apply this to IMPT-514, which is the same product as IMPT-314 on the heme malignancy side? This is our autoimmune program. So I won't go over this data. Ben has already gone over it. Phenomenal data, first in Nature Medicine for the first 5 patients, and now an update in the New England Journal of Medicine. Now, I don't believe every single construct and therapy will replicate this data. It's just, this is pristine data, and this is a compassionate use program. And one has to realize that when you go into a multi-center trial across multiple investigators where the choice of patients becomes more and more diverse, you're just going to see some variability. So you want to make sure that the patients that are targeted for these types of therapies are the ones that are going to get maximum benefit. And I think there are a lot of patients in that category. So we are focused, just like Kyverna is, on high unmet medical need. We are looking at a differentiated safety and efficacy profile. The tolerability profile of this makes it very likely outpatient therapy. And I just want to point out, in our heme malignancy program, we already have the option for outpatient therapy, just started. So imagine cancer patients are getting this CAR T cell therapy at a much higher dose than the autoimmune side as an outpatient. So we believe outpatient therapy is one of the key table stakes for the autoimmune side. My wife is an autoimmune patient with moderate to severe disease, and she gets her monthly injections on each thigh as an outpatient at our home. Okay? And I think that's what some of the expectations will be when we get into these broader markets. And we have multiple data readouts coming out through 2024 and into 2025, both on the heme malignancy side and autoimmune side with the same product. At a lower dose, by the way, on the autoimmune side, because you just don't need as high a dose as compared to the heme malignancy side. So why CD19 and CD20? You just heard about broad coverage of CD19. But what we have seen in our hands and what the literature has reported is that there's quite a bit of variability in CD19 expression. And when we get these donor leukopaks, we also phenotype for their various B cell compartments, and we also see this variability. What you see quite consistently is CD20 is a highly expressed, high-density antigen, sometimes 5 to 10 times higher than CD19. And remember, the construct is a OR-gated CAR. That's Boolean logic for saying you can bind either 19 or 20, and you will activate the CAR T cell. So when you have a high density of any one of these antigens, these particular CAR T cells get activated in this process. So you don't need a very high dose as a result. We also don't need a long manufacturing process, because what you're seeing is that you're able to make dose very rapidly in the autoimmune patients versus in the heme malignancy patients. So we're looking at a shortened manufacturing process also for autoimmune. But very importantly, if you look at the B cell phenotypes, inflammatory age-associated B cells, which are tissue-resident B cells that produce a lot of autoantibodies and that lead to immune deposition disease, have very high levels of CD20. And you want to make sure you get to those target organ sites. In addition, what you're seeing is sparing of the long-lived plasma cells, very similar to what Kyverna is aiming to do because you want to protect vaccinal immunity. And last but not least, there's a subset of dysfunctional T cells that express CD20 on the surface. Now, I'm a B-cell immunologist, and I always thought CD20 is a B-cell target. But why do these T cells express them? We don't know the exact mechanism. It could be trophophagy, or it could actually be primary mRNA expression, and therefore protein expression. That's being worked out, but we're clearly seeing this population in our own hands. We're seeing cell types with T cells with CD3 co-expressing CD20. And these cells are actually producing a huge amount of cytokines, pro-inflammatory cytokines. And most of these autoimmune diseases are complex diseases. They're not just B-cell diseases or T-cell diseases. There is an interplay in the immune compartment. And I'm hoping that by targeting both dysfunctional B cells and dysfunctional T cells, you get the patient a longer benefit. So what have we seen in our hands? High transduction efficiency, good fold expansion and making dose, B-cell aplasia, IgG secreting B cells also are targeted, but most important is the right-hand graph. Remember I told you in the cancer side we are seeing just lower interferon gamma production. In each and every one of the various indications we have gotten donor leukopaks, we're seeing the same. Our construct and cell type in general is associated with a much lower gamma interferon production, and that is one of the reasons we think that you're not seeing the CRS that you would see typically with other products. And we have not seen ICANS in the UCLA study, and hopefully that'll continue with the lower B-cell burden, antigen burden. So this is our program. We're doing a combined phase 1-2 study, the study is right now in the phase 1 portion. We already have a target dose identified based on the heme malignancy side. We will expand that to non-renal severe SLE. And then the registration pathway still has to be worked out, but we believe that the profound impact of these types of therapies gives you the opportunity to have single-arm trials in many areas. We're also starting the basket trial ex-US with 3 autoimmune indications and then filing additional INDs in the US. And remember, we just got into this area in the fall of 2022 and with a team that was focused on oncology, so we've moved pretty rapidly. We also got an $8 million grant, a competitive clinical grant from the California Institute for Regenerative Medicine as a validation of this particular work, and that grant actually supports the lupus nephritis SLE study. So we're addressing therapeutic gaps. We're designing our products for durable and safe outcomes. We have exceptional phase 1 data, robust and scalable manufacturing, and a lot of clinical readouts coming out through this year and into next year. Thank you very much for your time. They think I need a lot of water, clearly.
Okay, a great start to the session. Thank you, Ben. Thank you, Sumant. I guess the biggest set of challenges that are facing the applications of CAR T-cell therapies to the autoimmune space are manufacturing and possibly safety, maybe not necessarily because of the area of application being non-oncology. It's not terminally ill patients that have failed all other forms of therapy that are now being addressed for this type of treatment, but also this substantially greater number of patients, as you indicated, 8 million as the target population. So what are your further comments? Maybe first with manufacturing, and Sumant, you referred to specific modifications to the manufacture of the CAR T-cell for applications in immuno-oncology. Maybe we can start with that and then move on to the safety issues and other challenges.
Yeah, so before I joined the company, it was independent of me, so a decision was made to put at least clinical stage manufacturing in-house. I'm not 100% sure, like I would have had the foresight, but it turned out to be a good thing, so I got to give credit to its due. I joined literally, signed the contract, I think the day they signed the lease. So there's no backing out. Okay, so at that time, I said, okay, I'm going to make it work because manufacturing is a key part of cell therapy. I lived 6 minutes away from the manufacturing facility because it was nothing. It was a shell. And I moved 6 minutes away because I knew manufacturing, if you don't do it right, you don't build the facility right, it can truly destroy the company. So the decision was made. I came into the company, and we created our clean room suites to handle clinical stage manufacturing, but we have the ability to also scale to some level of commercial scale. I don't think we're going to do that forever, because I think we do need to partner with a CDMO for large-scale manufacturing. But what we're trying to do is shift from our current process, which I would call semi-automated, to as fully automated as possible, and then do the tech transfer as needed. And that's, I think, important, because what you see in cell therapy manufacturing is that there's a lot of requirement for skilled labor. You can't just take someone and 3 months later they're just doing this work. There's so much attention to detail on both the manufacturing side and the quality side. You want to automate so the human variability is reduced in the manufacturing process. We have a really good lentivirus source, a very good plasmid source, all of that is fixed. What we don't want to do is to say, we're going to scale up to tens of thousands of patients in a semi-automated process. That is when you cannot serve the market. Okay, and that's one of the things that we're focused on right now. In the second half of this year, we're hoping that we'll switch over to fully automated process. And we already have a system that we've been working with, and with our process, it works extremely well. So we're quite positive about that. And the other thing you want to do is you want to make those changes before you enter into phase 2, because you just don't know what subtle changes in manufacturing could do to the product at the end. And then we're also working to shorten the process. We started with a 12-day process. We're now down to an 8-day process, and we're even getting shorter on the autoimmune side. The point of this is that how can you scale manufacturing for the large number of patients that are going to come through this funnel?
And maybe you could comment on this also, but also in particular whether the issue of using peripheral blood versus leukapheresis material poses any particular interest, challenges?
Yeah, that's technology we're working on currently, and we're I can't talk too much about the details about the technology that we're using for that. I did want to second Savant's comments around how important it is to focus on manufacturing. We did choose the CDMO route. Karen Walker, who's a big advocate here at ARM, is our Chief Technical Officer, and so she has multiple decades of experience in the manufacturing space and quality space as well. And having a consistent manufacturing process is critical, and down to each individual operator. You know, I just saw a thing from Charles River recently around how operator to operator variability was even greater than patient-to-patient variability. And so that's critical to get your manufacturing right. We have an advantage in autoimmune patients in that they do have healthy, robust T cells to harvest, whereas in oncology patients, they're pretty beat up from all the chemotherapy they've had. And so we're not seeing manufacturing failures. We're not seeing the type of variability that you might see in the oncology space, which is a real advantage. It helps us accelerate our PQ lots and dealing with all the engineering runs and everything. We have a reliable seed stock to work from, which is each individual patient for the autologous. And so then to pull in whole blood really helps the patients out, because you're avoiding 4 to 5 hours of apheresis, and that's not fun. But you're also being able to, that does put some more demands on your manufacturing in terms of what your cold storage, your supply chain, everything else is. So it's not painless.
Right. So maybe following on from these aspects of manufacturing, would you maybe both, Ben, you start with address the issue of autologous versus allogeneic, because that also has a huge impact on availability of material for patients, ease of treatment, et cetera, but I have a couple of biological issues with some of those approaches. So, Ben.
Well, I really appreciate the elegance of autologous cell therapies in that you're your own cure. You're helping yourself. you're getting these cells harvested from you, and then they're being modified, and it comes right back to you. And so it's such a simple concept, and so elegant from a medical perspective. But that isn't scalable on a global perspective. And so to be able to access markets around the world that don't have autologous manufacturing facilities and wouldn't be able to do the apheresis that would happen in our first program. The allo donors are really going to be what allows Kyverna to go to a global marketplace cost effectively and everything, because the allo program will be much more cost effective as well, because it's just a much more efficient, a single donor can supply 100, 200 doses. So,
Suman, would you comment on the issue of allo versus autologous, and I'll come back with some additional thoughts for you guys.
Yeah, so I think anything you can go to get off the shelf would be great. So let me say that. I just mentioned my wife, she's an off-the-shelf product, right? It's a biologic. But what matters to the patient the most is that they have durable clinical remission from their disease. If my wife stops her injection or misses a dose, her psoriasis comes back with a vengeance. Okay? And what I want to make sure, like, we're all testing for potential for allo, and I think that's the right thing to do. But it'll come down to durable clinical remission in these patients. That's all it'll come down to. When I joined in end of November 2021, Impact Bio, I was facing an uphill battle people were saying, allogenic therapies will eat you for lunch on the autologous side in heme malignancies and oncology. It will happen, and it was hard, okay? Thankfully, we were able to raise our Series B, and 2022 was a hard year because that's what you faced. And then ASH 2022 happened in December, and not a single person mentioned allotherapies to me. Because at that moment, everyone realized that the allotherapies were challenged in the heme malignancy and oncology space. I'm not saying it's going to hold for autoimmunity, but I think it comes down to generating the evidence. And we have interest in allo also. We're looking at a couple of platforms in this particular area. But we want to make sure that if you take a CAR construct like a CD19, CD20 into the space, you make sure you choose a platform that has the potential durability of clinical response. We're also looking at in vivo and to see if that's a possibility. And these are early days of where you can put this. I'd like to get to off the shelf. So allo is not a topic anymore as much on the heme malignancy side. I think the time will only tell what kind of clinical durability you get. But I'm very glad we're testing it to get that answer, okay? And we are wondering when is the right time for us to step in that direction. Because we have the technology on the CAR side. The question is, what is the cellular technology you want to bet on?
Well, I can certainly see the attractiveness of allo CARs in immuno-oncology. But I have a problem with thinking of allo CARs in autoimmune disease, given the duration of survival of the allo cells in the patient and potential need for ever-long expression of the immune suppressive function. Any thoughts on that?
Yeah, I definitely have thoughts around that. I think that what we're seeing in the clinic is that a sharp, severe depletion of B cells is really an autoimmune reset. It's if, you and I would probably remember the blue screen of death on a computer. Really what these autoimmune patients are experiencing is this spinning of just their immune system not understanding that there really isn't a problem. And just like a computer can just get hung up and spun, but if you just put that little paperclip in the little hole there and you reset the computer, it shuts down, comes back up, everything's fine. That's what we're seeing in the clinic, is that if you get a really severe B cell depletion, then the patients can replenish those B cells back again without that autoimmune trigger. and so you really can see a persistence of durability, and that's what Dr. Shutt's patients are out over 1,000 days with no resurgence. So we don't see the real need for a long persistence. So allo should theoretically work just as well as an autologous from that perspective.
So Matt, your thoughts on this?
Yeah, so this is a science lab session, so forgive me if I will debate this one a little bit, okay? We should be debating this. That's where debate should happen so that we understand the science behind this. So B cells have been targeted by anti-CD20 therapy for a very long time. You can show deep, persistent peripheral depletion of B cells, but yet you don't see that. The patients have long-term complete remission. Why is that? And by the way, on the CD19 side, it's the same. In NMO, you see actually deep CD19 B cell depletion, but yet you see disease acuity come up in some patients. And part of this is due to the fact that cells, B cells, T cells, whatever cells, they're not in the periphery only. Mm-hmm. They're in tissues, they're in germinal centers, they're in the CNS, they're in the testes, the ovaries. They're there for a reason. They perform an immune function. When they get dysfunctional, they are going to be in those resident tissues. And I'm not saying that allo can do that job. What I'm saying is we know that you need some level of T cell, let's call it effector function. Okay, whether it's a T cell or it's a NK cell, it's just a cell, right? You need that persisting long enough to be able to get those dysfunctional cells where they're sitting. And I think that's an important concept, and that's why I said only time will tell what type of clinical durability you get.
I guess the bottom line is that we don't have all the information right now, and there is the possibility of resetting the clock altogether, rebooting by taking the B cells out. And at another level, as you hinted during your presentation, it matters precisely what B cell has been taken out, possibly, and what other cell, in addition to the B cells, have been taken out. So how much of the drivers are taken out for the efficacy of autologous versus allo? I'd like to maybe switch for the next 10 minutes or so.
Before we move on, as Samat was talking, there was another point that I would like to make is coming from a gene therapy is where a lot of my previous work was in. It was a one and done. You like for an AAV gene therapy, you'd get it once, and if you're going to try and redose, it's not going to work, at least not with today's technology. These CAR T cells could be redosed. And so if you do see a resurgence of disease 5 years later, when I came in to focus on cell therapy, I was thinking that's the end, that was our only choice. But if you can redose, and especially if COGS are down with allo and everything else, then you're actually able to get a maintenance over time.
Yeah. That's a very good point. It depends when redosing becomes necessary. If it's on a monthly basis, it would be out of question. Exactly. If it's 5 years down the line, then of course it's different. Now, switching to the safety issue, my group at ViraCell has produced 200 batches of lentia and gamma retroviral vectors for clinical trials, primarily in CAR T cell space. And we're aware of the issues that are surrounding the potential safety of gene therapy. In immuno-oncology, I think it is a big enough issue, and in autoimmune treatment, of course, an even bigger issue. I think the question of safety switches is particularly important. ViraCell is putting a lot of effort in this space. What are your thoughts on that? How important is it to have regulation, inducible or autonomously controlled in the therapeutic gene expression and or against malignant transformation or other, indeed, side effects that may come along, overexpression of some of the payloads within the vector or within the target cell that may be of seriously undesirable side effects. So safety, Ben, and then Samantha.
Safety first, always. We're not seeing the overexpression. You know, the issues that we're really trying to mitigate are CRSs and ICANS. Those are the obvious CAR-T safety risks. And there's enough history in the oncology space to really be able to manage the patients in an effective way. And we're just not seeing a risk of having overexpression pop up in the way that you may see in other modalities of treatment. You know, the use of tocilizumab and other mitigating treatments to help kind of cap off the T cell expansion, because in some patients you do get high T cell expressions. And so we just need to be able to mitigate that, and that is really our primary concern.
Okay, so those are side effects of the therapy that are associated with responses in terms of something like overexpression of cytokines. How about insertional mutagenesis and issues of that kind? And particularly important for me is the fact that we are now talking about 8 million as a target population versus a substantially smaller, several 100,000 as a target population.
Yeah, Samant and I were talking about that before.
Yeah. So there's, in the autoimmune space, any patient who's chronically immunosuppressed, you're just going to have a higher baseline risk of malignancies, including lymphomas and other malignancies, as well as infections. So the most important thing in any development of any therapy is to get the signal from the noise in the system. They're just going to have a higher level. And I think what we want to do is have a very rational mind in this particular situation. I think, this is my opinion, I think the FDA did the right thing by making a class label very recently around the potential for CAR T cell associated malignancies, because the package insert is meant to educate the patient, okay, and the provider who will educate the patient. And that class label, until proven otherwise, will stay. And I think it'll also stay into the autoimmune space. That doesn't mean, oh my God, you know, patients should not be getting this. You have to think about the benefit to the patient and the potential risk to the population. This was not discovered in several patients. It was discovered after years of therapy. Okay, it was seen in a subset of patients. And if you look at the biologic medicines that are given today for psoriasis, which I'm quite familiar with because of my wife, I've looked at it, it has a black box warning for malignancies and infections. And I think that is going to be the accepted class label until a specific product proves it otherwise. Now, that being said, if there is regulatory acceptance for a safety switch, right now there's some hesitancy to put in additional packaging into a cell, okay, just for the sake of maybe something may happen, and therefore you could give something that could kill the cell. That acceptance is not there fully yet, okay, for the maybe. But if it turns out that it's a good thing in the future and it's quite safe, it doesn't really alter the product, could be in the next decade or so. The rates are not high enough to mandate something earlier, because the benefits far outstrip. If you see the Schett data and you're seeing the early Kyverna data, I mean, the benefits are just unbelievable compared to the potential for risk.
Yeah, and that's, just to follow on what we took the secondary malignancy, you know, letter that everybody reacted to from the FDA at the end of last year. We took that very seriously and actually met individually with the FDA to get clarity on what data could they share with us, and Peter Marks publicly discussed it at the ARM meeting at JP Morgan, where he did not see the risk-benefit being altered from the data that they've been seeing. So the agencies are very supportive of the work that we're doing in the autoimmune space for all of the CAR T therapies that are going into autoimmune. You know, Intel data proves otherwise.
Well, I agree with that overall assumption, but also think that biology stands at a very exciting point right now, where it is possible to generate programmable vectors that have got multiple response elements built in the same way as you build in digits for a telephone number, and therefore be able to respond to a whole variety of, if you like, physiological cues and signals, including some of the safety requirements in substantially better ways in the future. And that is what we're working on at Viracell as one of the main points of focus. So that kind of approach for safety may have other side effects of a beneficial kind in terms of better regulation, in terms of making sure that for instance, immune suppression is not across the board indiscriminately throughout the body. If an autoimmune disease in some other tissue needs to be suppressed, maybe it doesn't have to result in similar immune suppression, say, in the liver, and have the associated risk of reactivation of resident viral infections and things of that nature. So what I'm saying is that I absolutely agree that there is a great safety profile out there, but also equally exciting and promising is the presence of these potentials for better programmable, regulatable vectors and target cells that may bring a degree of efficacy as well as safety that is completely unprecedented. Now, we have about 6-7 minutes left. I want to see if there is any burning questions, comments in the audience. Right over there.
Madan Jagesh, Obsidian Therapeutics, fantastic discussion. We have taken the lymphodepletion from the world of cancer, allogeneic transplant, called that LD for CAR T cells. As you speculate forward, do you foresee a product free of lymphodepletion, especially in the world of autoimmunity? And a second slightly different question, how are you thinking about potency assay for CD19 CAR Ts for autoimmunity versus malignancies?
Great questions. In that, I think, let me take a stab at this, and then maybe Ben has some thoughts here. But I do agree that lymphodepletion, if we can go away. So we have reduced the intensity of lymphodepletion for the autoimmune patient. We also have reduced the lymphodepletion for the CAR T experienced population versus the CAR T naive population, just because their bone marrows are probably a little bit heavier beaten up. So we have this reduced intensity. Ideally, you would like to go away from potent lymphodepletion. So the combination of cyclophosphamide and fludarabine have a very interesting mix. If you take away fludarabine, too much fludarabine, you don't get this cytokine milieu that allows for appropriate CAR T cell expansion. So if you want to replace it with something, it's not just about knocking your cells down so your CAR T cells can expand. It has to create an environment for those CAR T cells to expand, which includes inherent cytokines being released by the body just to stimulate hematopoietic stem cells to start replenishing the body. And the bystander effect is the CAR T cell benefits from it. So whatever the regimen is, people have talked about antibody depletion and other ways of doing this. The challenge is you still need to create the right environment for your CAR T cells to expand, because what you saw in the curves that Ben showed and that I showed, you want it to peak and you want it to start dropping off, because you want the antigen to be depleted, anything carrying that antigen. So we are looking into this. The challenge is that there's not been any proven evidence, but I do believe when you talk about like safety, acute safety, that's one that we should be looking at. We should be looking at options in that area. Mm-hmm. The second question you asked, I'm so sorry, maybe Ben remembers this. It was more of a regulatory question.
Yeah.
What was the second question? Apologies. Madan? Oh, yeah. Potency assay. Everyone's nightmare sometimes when you have a potency assay. What people have found with gamma interferon is that it's kind of a very highly variable potency assay. Even the commercial products struggle. Patients are going to be very, very different and in this particular area. And then you have the cytotoxicity assay. And any biologic assay has just a very different variability than even gamma interferon is very variable, and cytotoxicity assays are quite variable. But I do think that if there's a way of getting the cytotoxicity assay, which is probably the most relevant, that you are doing functionally what you're supposed to do, which is kill the target cell. I think if we can start locking that down as early as possible, it gives you a better handle of how effective your cells are for that particular patient. Doesn't mean that the cell will expand in vivo the same way and kill the same way, but at least in vitro setting, you get a good sense for a release assay. By the way, we not only do our own manufacturing, we do our own quality release. And actually that has allowed us to start standardizing things very, very early on. And our turnaround time has been very competitive in the heme malignancy space. I think in the chronicity of disease for autoimmune, you have a little bit more luxury in terms of time, but it's always good to get the product to the patient sooner rather than later.
Yeah, if I could advise any early stage biotech, it's get your potency assay done, and just get that clarified, and that'll solve a lot of your other questions along the way. Yeah.
Yep.
Yeah.
We have just over 2 minutes with a very sharp break.
For the first half of the question, lymphodepletion is critical, but it's also it's unknown how much it's really needed for the autoimmune patients. And so we are experiencing, we're experimenting with different lymphodepletion ideas, and we will be exploring that. There's such a big unmet need, and there's so many opportunities with so many patients that are suffering today. And there's irreversible damage that's happening to millions of patients today that we could be helping. And so doing what's known with a standard off-the-shelf manufacturing process, a CAR T that we already have a pretty good safety profile understanding of, lymphodepletion is critical, and we'll work through those issues. And it's actually what has been seen as adverse events that are popping up are most likely related to the lymphodepletion. But we just need to get out there, and the risk-benefit is just mind-boggling. I haven't seen it in the monoclonal antibodies and the gene therapies. This is such a clear win. Yeah.
That's wonderful. Obviously, gene therapy needs to be able to do more than is achievable by conventional pharmaceuticals. And therefore, it is very encouraging to have heard the presentations from Ben and Sumant this morning, particularly your inspirational movies of the patients after their treatment. Really terrific bundle of hope for the future. Thank you. Thank you for being such great pioneers.